Which came first, the chicken or the egg? DNA or proteins? DNA needs proteins to be made, but proteins need DNA to be synthesized. If you asked which was discovered earlier, that would be proteins. It wasn't until 31 years later where DNA was finally discovered. But before I can tell you which one existed first, you need to understand this. For years, scientists thought that proteins carried genetic information. They were just more complex, more powerful.
Thinner. Look better.
And DNA was just a simple, boring molecule. But in 1944, scientists realized that this boring molecule actually contained all the genetic information for life. So we began to think, how does DNA make proteins? And it all starts in the nucleus. But DNA can't leave the nucleus, so it sends a messenger that carries a message called messenger RNA, aka mRNA. The process of making mRNA is called transcription. This is a gene. It's a section of DNA that codes for a polypeptide. RNA polymerase unwinds and unzips the gene to be expressed by breaking the hydrogen bonds between the complementary bases. RNA polymerase uses a template strand, aka non-coding strand, as a template to assemble the complementary mRNA strand. Now, where the hell does it get these nucleotides from?
Where, Dwight?
Seems like you already know where.
They're literally just free floating in the nucleus. Since we're here, I might as well also explain what a nucleotide is. A nucleotide is a unit made up of a phosphate, sugar, and a nitrogenous base. By the way, nucleotides make up DNA and RNA, which are both nucleic acids. Just gotta put that out there. Students always confuse coding strand, non-coding strand, template strand. I'm gonna explain it once. Coding strand is a copy of the mRNA, but it's got a uracil instead of thymine.
Who are you?
You.
No, not me, you.
Yes, I am you.
That is because the RNA polymerase needs to use the template strand or the non-coding strand as a template in order to produce the mRNA strand. So in order to make it the same sequence as the coding strand, it needs to use the non-coding to make the complementary of that, because the complementary of non-coding is coding. Anyway, so if you can't remember any of that, just remember coding is the copy. Keep in mind, in eukaryotes, a gene is made up of both coding and non-coding DNA, meaning some are directly used for polypeptides and some aren't. 98% of DNA in eukaryotes is non-coding DNA. That is why this premature mRNA needs to undergo RNA processing. During RNA processing, 3 things happen. The first involves the removal of introns. An enzyme called spliceosome will remove the non-coding sections of DNA that are in between coding DNA. These non-coding sections are also called introns because they are intervening sections of DNA. When my students can't remember which section of DNA gets removed during RNA splicing, I always tell them, introns go to the bin. mRNA is also very fragile, so a 5' cap and poly-A tail are added to prevent degradation once it leaves the safe nucleus and into the cytoplasm. After leaving the nucleus through the nuclear pores, mRNA rushes to the ribosome before it starts to degrade. At the ribosome, the mRNA is used as a template to produce a polypeptide chain, which is a chain of amino acids. The mRNA binds to the ribosome at the 5' end, and it essentially becomes sandwiched between the large subunit and the small subunit.
What are you?
An idiot sandwich.
Idiot sandwich what?
An idiot sandwich.
But the ribosome can't bring in the amino acids to form the polypeptide chain. So instead, tRNA does that. Who the hell is tRNA? tRNA is a 3D RNA molecule that carries 3 nucleotides at one end called anticodons and carries an amino acid on the other end. Students always mess this part up. So the tRNA anticodons temporarily bind to the mRNA codons. The ribosome facilitates this by holding them in close proximity. As soon as this temporary bond forms, the amino acid detaches from the tRNA and begins the polypeptide chain. Here you can see a tRNA carrying methionine attaching its anticodons to the mRNA codon. As soon as this temporary bond forms, methionine detaches and begins the polypeptide chain. We now see another tRNA carrying proline forming a temporary bond with mRNA's codon. Do you notice how the anticodons are complementary to the mRNA codon? This is exactly what allows them to form that temporary bond. So proline detaches and the ribosome allows the formation of a peptide bond between the proline and the methionine. This process repeats. Keep in mind that only 2 tRNA can attach at once. One would just be leaving and the other one would have just arrived. Again, a tRNA carrying glutamic acid temporarily bonds, the amino acid detaches and joins the chain. The empty tRNA leaves while another one comes in. And finally, a stop codon is reached. But keep a note that the stop codon is not an amino acid. It's literally just an indicator to stop translating because that is the end of the mRNA strand. And this produces a polypeptide chain, a chain of amino acids bonded by peptide bonds. But making a polypeptide isn't the end. It's actually just the beginning. The next part differentiates one protein from another. That chain must now fold because structure determines function, and there are 4 ways that a protein can fold. They can have a primary structure, which is literally just that sequence of amino acids in a chain like beads on a string. Or a secondary structure where they form hydrogen bonds between the chain to form either alpha helices like a curly fry or beta pleated sheets. Or a tertiary structure where the entire polypeptide chain folds into a more complex 3D shape, or the most complex, where it produces a quaternary structure with multiple polypeptide chains combined together, like hemoglobin. This leads me to ask, what the hell did we just make? This was just a section of a complex hemoglobin molecule. Of course, it's not 4 amino acids long, it's actually 574 amino acids long. But for obvious reasons, I'm not gonna show 574 amino acids. I've actually created a simplified model to show what hemoglobin would look like. Hemoglobin is actually made up of 2 alpha helices and 2 beta pleated sheets. So that is actually what I have right here, and you can see there are white pom-poms on each one. That is actually because there is an iron that is found on each chain. That is why if you guys are iron deficient, which I think every single female is iron deficient, you would also have symptoms of anemia. What if I just change this one base? What if I change this one adenine to a thymine? What ends up happening to the actual polypeptide chain? The codon actually goes from GAG to GTG. Now, you might think this is just one base out of thousands. Like, what significance can this have? Well, number one, it would actually change the amino acid. So it changes the amino acid from glutamic acid to valine. Imagining this is the polypeptide chain. Of course it's not. Imagine one amino acid out of like 574, one changes. What significance is that gonna have? Well, do you guys remember when I told you that structure leads to function? Well, you've just changed its structure. The hemoglobin ends up misshaping because of incorrect bonding and folding because of that one amino acid. And hemoglobin makes up red blood cells. So what happens to the red blood cells that carry this misshaped hemoglobin? Well, they go from being a biconcave, flexible shape to a rigid, sickle-shaped red blood cell. So the way that I imagine it is a red blood cell with normal hemoglobin, looks like a donut without a hole in it, whereas a sickle-shaped red blood cell kind of looks like a rigid banana, like a frozen banana. Well, this one base that you changed actually caused a genetic disease that was responsible for 34,400 deaths worldwide in 2021. Sickle cell disease. So rather than being flexible and moving easily through the blood vessels, these sickled cells rupture and block blood vessels. Rather than carrying 1 billion oxygen molecules at capacity, these sickled cells carry less than 50% of that. Don't worry though, we are working on a solution to edit that specific gene using CRISPR editing technology, but that's for another video because I think I've definitely gapped enough today. Anyway, so what came first, DNA or protein? It was actually RNA. So we actually figured out that it was RNA that most likely existed first, even though we pretty much have no like solid evidence. We don't, it's not like they were existing back then. I know this is out of the HSC syllabus, but the RNA that was actually discovered to have, possibly originated life is an RNA molecule called ribozymes. So they can actually catalyze reactions and produce proteins, but they also carry genetic information. Anyway, so let's not talk about ribozymes. Instead, I've actually got 2 questions that I put in the description below, an easy protein synthesis question and one very difficult protein synthesis question. So you should be able to answer both, but I've just included both of them to give you a bit of an understanding of where you are sitting at. I've also put the marking criteria for both of them so you can actually mark your own work and you can look at the sample answer and see if there's anything that the sample answer says that you don't, so you can add that in. So remember, like and subscribe. Anyway, I'll see you guys next lesson.